if students have chewed gum or like maybe eaten a big meal like right before this, it could affect the results.
So we should probably let people know right before, especially for right after lunch.
Maybe eating is not as big of a problem, but chewing gum, or I think I don't know if it's the artificial sweetness in gum or something about chewing gum. Anyways, pour a little 0.9% saline into a cup.
The exact amount doesn't matter.
The exact amount doesn't matter, but I usually go smaller because no one wants to drink or like swish around that much saline. And then bottoms up. 30 seconds of swishing. That's probably around 30 seconds. Alright, so label our Eppendorf tube. And Jed's pipetting 1 milliliter of our saline solution with our solids into the Eppendorf.
Amongst these tubes, we have an alt number, right? I think there's, can't see, is it 18 divided by 3 is 6, so 1, 2, 3, 4, 5, is it there? 2, 3, 4, 5.
Yeah, and I really like how Jed put the
attachment.
Yeah, the
lid attachment on the outside.
Yep, because that'll make it easier for us to get our cells out when we need to.
It's up there, and so what are we going to do? Just 1 minute, or
yeah, like 90 seconds.
do you want to put it at 14,000, or?
Yeah.
All right, let's take the tubes out, and I can take mine out. Let's see if I can even find a pellet. Oh, I can see a pellet. There's a pellet there.
Hey, look at that.
All right, let's see. Courtney, yeah, we have a pellet there too.
All right.
There we go. Yeah, pellet again.
Oh, nice big chunky pellet.
All right.
Lodged on the bottom, so you can just open your tube and pour the supernatant and like give it a little shake, and you're going to be left with a small amount, maybe like 50 microliters left that you just can't get out from pouring. Yeah, that looks good.
Right there.
Yeah.
In there.
Nope. So you can tap it to resuspend. Sometimes I like, if it's really chunky, I'll use a pipette, and I just go kind of up and down underneath the surface, and that can resuspend your pellet really well as well. Or
You can do that.
So next one,
yeah.
all right, so we're going to get the PTC primer.
Okay. I brought these beads. I knew what we were gonna do, so with the same letters, initials that we have here. So and sometimes smaller Sharpies work better if you have them, but we can make this work. So.
Bam.
We can also, it never hurts to write, once you put it in the PCR machine, to write the order that it is in before it goes in, so that then because sometimes it comes off because of the heat, so that way you're kind of guaranteed to know what's in it. So in the beads, the freeze-dried pellet has the TAC enzyme for amplifying your DNA, and then the buffer that it needs to run in your magnesium chloride. And then in your primer solution, you have your primers for the PTC gene. And then we're gonna add the DNA to it from our cells, and that's what we need to run a PCR. Your primer solution also has the loading dye, which is dye that is used for gel electrophoresis. So this allows us to skip a step later on when we're running the gels. We also have DNTPs for amplifying the DNA. So it should have everything except for the primers and the DNA.
I always make sure that the bead hasn't popped out.
Yes. There we go.
And are you okay if I just
Go for it.
put it in every one? And I can use the same tip because there's nothing different between the different tubes, right? I'm going to let it run down the side of the wall. Six of them.
I like to just kind of close them up and actually mix the primers in before adding the DNA, just to get the reaction started. And all you have to do is make sure that the pellet dissolves. So you can kind of see it's bubbling a little bit, and you won't be able to see the pellet anymore. So one thing, you just kind of tap it, and eventually it'll go clear, and it'll look like it's fully mixed.
We should put these lids on then.
Yeah.
so we don't lose anything. Can't really see very much liquid moving.
Yeah, kind of hard. We can also do a quick vortex
Just
or tap before we do the PCR. Yeah, it looks like those are started, so that should be good, and we
pretty
could
good.
add the DNA. And then I think for students, especially when we're adding the DNA, the primer is easy because it's dyed, right? It's pink, so it's
Yeah.
easy to tell that you've gotten it in there. The DNA, we're doing a very small 2 microliters, and it's hard to see. So I think the best thing that you can do when you're pipetting is to hold the tube up at eye level and actually watch the DNA leave the pipette tip and go into the test tube. Because the key here is that we're going to amplify their DNA. So if they don't get DNA into the PCR tube, they're not going to get any results. So I always lift my tube up when I get the DNA out. So we're just getting DNA from our cells. So we take the cells out, and then I watch it go into the test tube. And I've been doing these for many years, and I even
Still
still
do
watch it go in because I think it's a very important step.
That's good. All right, so 2 microliters. So let's see, I have my cells in here, and probably should mix these up a little bit again. Make
Yeah.
sure everything there. And hold it up, and make sure I have some liquid in. I'm not sure if you can see that liquid or not. And I was on the first one, right?
Yeah.
All the way into the liquid. I usually pipette up and down a couple times.
For sure.
Got my first one done. Close my lids. Mixed up.
I'll mix my cells. And then, again, I'm gonna pipette and watch it go into the pipette tip. Very small amount of liquid. And then I'll find my test tube and I'll watch it go in and down, and then I'll go up and down to kind of mix it. But making sure that all the liquid is out.
Is every level?
Yeah, and you should balance them, but.
Do as we say, not as we do.
How do you run these?
I just turn it on. There it goes.
You only need to do like 2 or 3 seconds, and because PCR is so light, it doesn't I mean, it matters, but it's not as detrimental if you don't balance it perfectly. So you should, it's okay if there's some air bubbles, but all of the pink should be at the bottom, and it shouldn't be cloudy. The beads should be mixed in now. Cool.
So we just turned on the machine. This is the screen that comes up. We're going to hit enter for registered user. Now we're going to find the registered user. We're using biology, BIOL. The right arrow, the bottom arrow. Then we hit enter again. Then we want to go to the protocol library, and we have a PTC program. So let's see where it goes. Here we go, PTC, enter that. And it's all programmed to go, so we should just be able to enter the protocol. Hot start. Do you usually use hot start, or you not worry about it? Put no, and then we use that default, and our volume is going to be, I'm just going to put 25 microliters because that's pretty close to where we're at. Enter there, and now we're ready to begin the run. So we hit F5 here, and there's the program.
And then we close ourselves, right?
Almost on. There's a heat block in the lid, and also a heat block down below, which prevent the liquid from condensing on the lid.